This conference will now be recorded.
Hello students. Now we are learning about biomolecules, which is a bit difficult chapter of biomolecules, but this chapter doesn't have that much weightage in NEET syllabus. But still the questions, whichever you are getting, that will be the direct questions from this chapter. So here we have already gone through the syllabus, like we need to learn chemical constituents of living cells, then the biomolecules, the structure of biomolecules, function of protein, carbohydrate, lipid, nucleic acids. Then about enzymes also we need to learn. So about enzyme, about the properties, enzyme action, classification and nomenclature of enzymes, all these things we need to learn. So here enzyme is a very important part. It's like a tricky portion which you can learn very easily. Okay, so now we are moving. Already we have covered carbohydrate, lipids and all. So now we are moving to the nucleic acid portion. See, nucleic acid means, we know that what is nucleic acid? That is the genetic material of all living organism, right? It's the genetic material of all organisms, and two most important nucleic acids which is present in the living cells, that is deoxyribonucleic acid or DNA, and riboxy deoxyribonucleic acid, we can call it as DNA in short form, and ribonucleic acid, RNA, ribonucleic acid or RNA in short we'll write. So DNA and RNA, these were discovered by Michel. Okay, this DNA and RNA, these are macromolecules, and these are made up of polymeric units which is called as nucleotides. So this is made up of nucleotides, right? So a nucleotide means that is composed of, nucleotide is composed of three molecules. One is phosphoric acid, then pentose sugar, then nitrogenous bases. Okay, so here phosphoric acid is there, then pentose sugar is there. Pentose sugar, that means the carbohydrate or the monosaccharide unit which is having 5 carbon atoms. Pentose sugar means they are having 5 carbon atoms. This may be deoxyribose in DNA or ribose in RN. That's why deoxyribose and ribose. That means this is the pentose sugar which is present in DNA, which is that deoxyribose, and in RNA which is present ribose sugar, which is the 5 carbon compound. Okay, next comes the nitrogenous bases. This nitrogenous bases, they are the heterocyclic organic bases which is having nitrogen. Heterocyclic organic bases, and that is having nitrogen in them. And there are 5 different bases which are categorized into two types. These nitrogenous bases are classified into two types: purines and pyrimidines. Pyrimidines. Okay, purines means they are double ring bases. Purines, they have double ring. And which are there are two purines, adenine and guanine. Adenine and guanine, they are the purines. Then next comes the pyrimidine, which is a single ring base. Okay, so this pyrimidines, that includes cytosine, thymine, and uracil. Cytosine, thymine, and uracil. Okay, so purines and pyrimidines. Purines are double ring bases, and which are the two purines? Adenine and guanine. A, G. A and G. We'll mention adenine with letter A and guanine with G. Then pyrimidine, single ring bases, which include cytosine, thymine, and uracil. And here, these nitrogenous bases. In this, DNA will be having adenine and guanine. Then cytosine and thymine in DNA. Adenine, guanine, cytosine, and thymine. Okay, but in RNA, this nitrogenous bases, they contain adenine, guanine, cytosine, and uracil. Instead of thymine, it is having uracil. And we said nucleoside plus phosphate group, that is forming nucleotide. So the nucleosides, they are considered as the smaller possible framework which is forming the unit of nucleic acids. Okay. So this is about generally, these are the main points which you are getting from the previous year question paper about nucleic acids. Okay, these many questions we are getting about nucleic acid from the previous year question papers. Next, we need to go for this DNA in detail. Deoxyribonucleic acid. So this deoxyribonucleic acid or DNA, this was first discovered by Misha. And from where he got this discovered? From the nucleus of pus cells, which is obtained from the discarded bandages in the Misha. This is from the pus cells he discovered, right? And he called this as nuclein. Okay, the outline. Next question used to come from the outline of Watson and Crick model of DNA. Okay, Watson and Crick model of DNA. Okay, so Watson and Crick model of DNA, which is the most accepted form, in that DNA molecule consists of two helically twisted strands. See, like this, there will be helically twisted strands. Okay, helical strands, and that is twisted. Okay, and these are connected together by this nitrogenous base pairs. Okay, these both helical strings or these strands, they are connects with base pairs. Okay, and these will align themselves in the anti-parallel or in opposite direction. This is, both the strands are anti-parallel to each other. Okay, and these two strands, these are intertwined in a clockwise direction. That means in the form of right-handed helix. Right-handed helix. Okay, and they have anti-parallel arrangement, we said already. And in each strand, we can say each strand that consists of backbone, which is made up of alternating deoxyribose sugar. Okay, backbone, deoxyribose sugar and phosphate. Okay, so the phosphate joins the two sugar through phosphodiester bond. Two sugars or two sugar units through which one? Phosphodiester bond. Again, about the nitrogenous bases, we need to say this nitrogenous bases, they are stacked inside the helical pair and they are with the base of the opposite strand through hydrogen bonds or H bonds. Okay. Then another special information, which is an extra information, different forms of DNAs are there. Okay. So in that, right-handed form of DNA is called as bDNA. Right-handed DNA form, that is called as bDNA. And this form, this bDNA is found in humans. Okay. And left-handed form is called as zDNA. Left-handed DNA is called as zDNA. This bDNA is found in human and zDNA that is found in nucleosome. Nucleosome. Okay. So this is two extra points and these also came in previous year questions. So you have to learn this many points about DNA. And next we are moving to RNA. RNA, ribonucleic acid. Okay. RNA, that is also one nucleic acid which is present in the cell, that is ribonucleic acid. So this ribonucleic acid is found mainly in the cytoplasm. In cytoplasm. Okay. This is mainly found in the cytoplasm and this is mostly present in single stranded form. Okay. So RNA is single stranded. Mainly it is single stranded. Most of the forms are single stranded, but in some retroviruses and in some kind of viruses, this is found in tumor virus, in retrovirus, tumor virus, in such type of viruses and all, this is in the double stranded form. RNA, you can see double stranded RNA. In which one? In retrovirus and tumor virus, you can see RNA in double-stranded form. And this RNA is of different types. There are 3 types of RNA, which is that messenger RNA. We can write mRNA, which is messenger RNA. Next, ribosomal RNA or rRNA. This is ribosomal RNA. And soluble RNA or transfer RNA, that is S or tRNA. Soluble RNA or transfer RNA, we can write S or tRNA. This transfer RNA and messenger RNA, these both are synthesized on the chromosomal DNA. Messenger RNA and transfer RNA, both are synthesized on chromosomal DNA. Okay, but ribosomal RNA is synthesized on ribosomal DNA of nucleolus. rRNA or ribosomal RNA, that is synthesized on ribosomal DNA. Ribosomal DNA of nucleolus. Okay, so this is about RNA, and all these are in the previous year question papers. So at least try to remember these many points. Minimum these many points you need to learn. Clear? Next, we need to move to enzymes, which is a very interesting topic. Enzymes. See, enzyme means that is a specific protein which is produced within the organism. Enzymes are proteins produced in the organism. Okay, and this enzyme is capable of catalyzing some specific chemical reactions. So this is having catalytic activity. That means it is catalyzing some chemical reactions, and they are having biological importance, they are having biological origin, and they will catalyze different reactions. That's why enzymes are called as biocatalyst. Okay, enzymes, these enzymes are otherwise called as biocatalyst. And here enzymes we can write simple enzymes are there, conjugated enzymes are there. Simple enzymes and conjugated. Conjugated enzymes are there. Simple enzyme means they are made of only peptide chain, so they have only protein part. Peptide chain. So only they have only protein part they have. Which one? Simple enzymes. What about the conjugated enzymes? Conjugated enzymes, they are composed of protein part and a non-protein part. Protein part and non-protein part. In which one? In conjugated enzyme. Okay, and this conjugated enzyme you can apoenzyme, then plus cofactor. That is the conjugated enzyme is composed of protein part and non-protein part. Apoenzyme, that means apoenzyme and cofactor. This apoenzyme, that is the protein part. Apoenzyme, we need to say that is the protein part of the enzyme, and this is very large in size. Then they are specific for an enzyme. Then comes these apoenzyme. This is thermoliable. An apoenzyme that doesn't take part in the group transfer. These many points, protein part that is called as apoenzyme, that is large and that's specific for an enzyme, and they doesn't take part in the group transfer. They are thermoliable. Okay, which one? Apoenzyme. Next is about cofactor we are learning, which is a non-protein part of the enzyme. Cofactor is non-protein part. Okay, and this is having small size. Which one? Cofactor. And this can be used in a number of reactions. Which one? Cofactor. And this is heat stable. Then involved in group transfer. Clear? And this cofactor next point we need to learn what is prosthetic group. Prosthetic group means the organic compound which is binding through covalent bond to a molecule. That means to a apoenzyme. Prosthetic group is the organic compound which is binding to the apoenzyme. Coenzyme. Next is the coenzyme. So what is coenzyme? Organic compound. Organic compound which is binding to apoenzyme which binds to the apoenzyme, and here vitamins are the essential component of coenzyme. Vitamins are essential compound of coenzymes. Next comes the metal ions. Metal ions form the coordinate bond with the side chain of enzyme and substrate. For example, we can say zinc, that is a cofactor for proteolytic enzyme carboxypeptidase. Okay, so like that, metal ion. For that example, zinc, which is a cofactor for proteolytic enzyme. And here some extra points which we need to study: NAD, NAD and NADP plus. These are the coenzymes of vitamin B3. Coenzymes of vitamin B3. Okay, vitamin B3, niacin. Okay, then FAD and FMN. These are the coenzyme for vitamin B2. FAD and FMN. These are the coenzymes for vitamin B2. B2. B2 is riboflavin. This is niacin. This is riboflavin. Next, we need to say about the term enzyme. This was coined by Kuhne. Then who isolated this first enzyme zymase from fruit juice? First enzyme is zymase from fruit juice. That was first isolated by Buchner. So these are the common factors which we need to learn about enzymes. Then we need to learn about the chemical nature of enzymes. Chemical nature of enzymes. See, all enzymes are globular proteins. All enzymes, globular proteins. See, except some recently discovered RNA enzymes, all are globular proteins. So some enzymes may be additionally containing a non-protein group. Some enzymes, they'll be having additional non-protein group. Okay, so they'll be having additionally they'll be having a non-protein group. So there are two types of enzyme on the basis of composition. So on the basis of composition, there are two types of enzymes. So we are going to classify the enzyme now. Classification of enzymes. Oxidoreductase. Oxidoreductases. This oxidoreductases, they take part in the oxidation and reduction reactions or transfer of electrons. Okay, so these oxidoreductases are further classified as oxidases, dehydrogenases, and reductases. Then another type is transferases. This transferases enzyme, these are the enzymes which is performing group transfer reaction. This oxidation reduction reaction, then this transferases enzyme performs group transfer reaction like a peptidyl transferase. Okay, next comes the hydrolases enzyme. Hydrolases. Here this hydrolases enzyme that induce hydrolysis. Hydrolases induces hydrolysis like amylase, lactase and all. Next, lyases. So lyases, they induce the cleavage. Which one? Lyases induce cleavage without hydrolysis, and addition of double bond is also taking place there. So example for lyases, aldolases. Aldolase, that is the example for lyases. Next, isomerases. See, isomerases, they help in the rearrangement of molecular structure. Which one? Isomerases. Rearrangement of molecular structure. So isomerase, epimerase, mutase, all these are the examples for isomerases. Next comes the ligases or synthetases. Ligases or synthetases. So these ligases or synthetases enzyme induce the bonding of two molecules after taking energy from ATP. So they are inducing the bonding of two molecules. Okay, so for that, from where they are taking energy? They are taking energy from ATP. So pyruvate carboxylase is the best example for ligase enzyme. So usually for naming of this enzyme. According to International Union of Biochemistry, enzymes are named by adding ASC. ASC. That suffix we'll add after the substrate, like lipase, amylase, maltase, and all. For that, we will add lipases, amylases, maltases, and all. Okay. Or chemical reaction like succinate dehydrogenase. Okay, so this is about the classification of enzyme. Next, we need to learn about the mechanism of enzyme action. Mechanism of enzyme action. First, the general mechanism of enzyme action, that is having two steps. Okay, so which is the two steps? Formation of enzyme substrate complex. First one is formation of enzyme substrate complex. This is the first step. And the second step is lowering of activation energy. Lowering of activation energy. These are the two main steps of enzyme action. So here we will learn about the first one, formation of enzyme substrate complex. See, when there is a substrate and one enzyme. So when an enzyme is acting upon a substance, one enzyme is acting upon a substrate or a substance, that forms enzyme substrate complex. That forms enzyme substrate complex, right? So this complex, this will undergo chemical change and the enzyme is regenerated in the later session. Okay, so enzyme substrate complex will form. Again, this is the from enzyme substrate complex, enzyme product will form, and from that enzyme plus product, this will get separated, product will be getting isolated. So two models has been put forward to explain the formation of enzyme substrate complex. First one to explain this enzyme substrate complex, that is lock and key model, lock and key hypothesis. To explain the production of this, different hypotheses are there. Lock and key model, which is proposed by Emil Fischer. This is proposed by Fischer, and according to this lock and key model, both the components, the enzyme and the substrate, have strictly complementary structure. Okay, they should have, enzyme and substrate should have complementary structure. That means how a particular key is fitting into a lock. So exactly like that, one particular enzyme should have a structure which is complementing the substrate. So exactly like lock and key, it should match. The complementary structure should be there for the substrate and the enzyme. Okay, that is one hypothesis for the formation of enzyme substrate complex. And next one states that induced fit model. Second one is induced fit model. Okay, in this induced fit model, which is proposed by Koshland, who proposed induced fit model? Koshland proposed induced fit model, and according to this, when an enzyme binds to substrate, there is a change in shape of the active site of enzyme. Okay, when an enzyme binds to the substrate, the active site means the site where the enzyme is binding on the substrate. So that active site, there is a change in shape in the active site of the enzyme that will take place. That's what it says. Induced fit model. It is inducing that enzyme to fit on the substrate. So there is an induced, it is making that due to that shape change, it is correctly fixing into that substrate. So like that induced fit model. So you should, from the title itself, you should be able to write, or you should get the concept. Okay, loc and key means it should be exactly matching, then only it will work, right? And induced fit means it is inducing, it is compelling to change the shape to make it fit, right? So in that way you should think. Next, first step is the mechanism of enzyme action, right? The formation of enzyme substrate complex, that is the first step. In the mechanism of enzyme action, two steps are there. First step is the formation of enzyme substrate complex. Then second step is lowering of activation energy. So enzyme substrate complex formed. Lowering of activation energy. This is the second step. Here, in all the chemical reactions, they have a potential energy barrier, right? There is a potential energy barrier, and this must overcome before the reactants can be converted into products. See, if that is overcoming this potential barrier, that reactants will get converted into products, right? So there should be an energy which is required to break this barrier that is equivalent to the activation energy. Okay, so if it is breaking that energy barrier, the reactants will get converted into products. Okay, so here what an enzyme does in a chemical reaction, what it is doing, enzyme, how it works, that reduces the activation energy, that lowers activation energy when forming enzyme substrate complex. When it is forming enzyme substrate complex, it reduces the activation energy. So afterwards, the energy level of the substrate that gets raised and that reacts faster way. Okay, so here when we are plotting a graph, here we are writing the progress of reaction rate, here we are writing, and in the y-axis what we are mentioning is potential energy. Okay, so here the substrate, there's a transition state to a product, right? Then it is forming the product. Here, this is a product formation. This activation energy without enzyme. This is the activation energy without enzyme. When we are using an enzyme here, that reduces the enzyme, lowers the activation energy. So here activation energy is getting lower, then it is forming the product. Okay, finally it is reaching to the product. So here activation energy becoming lower. So activation energy with enzyme, that is low, enzyme is lowering the activation energy. Okay, when this substrate S, then we need to learn about some factors which are affecting the enzyme activity. So which are the factors which is affecting the activity of an enzyme? So the activity of an enzyme can be affected by a change in the conditions which can change the tertiary structure of the protein, right? So first factor, that is substrate concentration. Concentration of the substrate. See, enzyme activity will increase with increase in the concentration of the substrate. So substrate concentration increases means that increases enzyme activity, right? So enzyme activity increases with increase in the concentration of substrate to a maximum level. There is a maximum level, right? Then it lowers. Then the factors which is affecting enzyme activity is enzyme concentration, another factor. Substrate concentration, enzyme concentration, we can say the rate of reaction will increase with increase in enzyme concentration. Rate of reaction increases with increase in enzyme concentration. When enzyme concentration is more, reaction rate also will be more. Then temperature and pH. Okay, so most of the enzymatic reaction, that rise of 10 degrees Celsius in the temperature, that double the rate of reaction between 5 to 40 degree Celsius. So enzymes are denatured at higher temperature because they are proteins. We said they are proteins. So enzymes will get denatured at higher, very high temperature means enzyme will get denatured at higher temperature due to their protein content or their proteinaceous in nature, and the rate of reaction will drop down due to the increase in temperature. And in the pH case also the same thing is happening. At very high pH, the proteinaceous nature that will lose, and that will reduce the reaction rate. Next, we need to learn about some other questions which came in the previous year question paper, that is about enzyme inhibition. Okay, enzyme inhibition, reduction or the stoppage of enzyme activity due to some adverse condition. Stoppage due to adverse condition, we can say, or chemicals, due to some chemicals, that is called as enzyme. Due to some condition, enzyme that will stop the activity, and sometimes due to some chemicals that will stop the activity. So this is called as enzyme inhibition. And the chemicals which interfere this enzyme activity, they are called as inhibitors. The substances which are inhibiting the enzyme activity, they are called as inhibitors. Enzyme inhibition, that is of different types. Competitive inhibition, non-competitive inhibition, and feedback inhibition. Okay. So these are the enzyme inhibitions, different types of enzyme inhibitions, which are they? Competitive inhibition, non-competitive inhibition, and feedback inhibition. This feedback inhibition, that is otherwise called as allosteric modulation. Which one? Feedback inhibition. So these are the main topics which you need to learn from chapter biomolecules. So we have covered all the important points within 2 hours of this chapter. So here in exam questions of this chapter, that is divided into different topics like carbohydrate, you need to learn protein, lipid, nucleic acid, and enzymes. So you will get, if you're learning this many points, you can answer almost every questions, because we have covered 10 years question paper now from this chapter. So there are 5 topics: carbohydrate, protein, lipid, nucleic acids, enzymes. In nucleic acid, DNA and RNA you need to learn very well. Then in enzyme you need to learn what is simple enzyme, what is conjugated enzyme, apoenzyme, cofactor, prosthetic group, coenzyme, then metal ions, then the chemical nature of enzymes and classification of enzymes. That you may get confused. So read it 2 to 3 times. In that, oxidoreductase, hydrolases, transferases, ligases, all these with example you need to learn. Then mechanism of enzyme action, in that lock and key model, then induced fit model, everything. Then the concept of activation energy with the help of graph you need to learn. Then change in the concentration of substrate, how that is affecting the enzymatic activity. So that you may get in assertion type questions. So be clear with that concept. Then about enzyme, how temperature, substrate concentration is affecting enzymatic activity, how temperature and pH is affecting, how enzyme concentration is affecting the enzyme activity. All those things you need to learn. Then about enzyme inhibition, like competitive inhibition, non-competitive inhibition, and feedback inhibition. All these 3 you need to learn. Okay. And about carbohydrate, be clear with the points of carbohydrate. And in nucleic acids you need to learn about the nitrogenous bases and pentose sugar, what are purines, what are pyrimidids, which are purines and pyrimidids, and how the pairing is taking place. Here adenine will pair with thymine and guanine with cytosine in DNA. Adenine with thymine, that is a double bond, and guanine with cytosine, that is a triple bond. And this is in DNA. Okay, and in RNA, one point which I need to say, instead of cytosine with uracil, it will pair. Okay, so these are the points which we need to learn from this chapter. Go through this chapter and make sure you are clear with all the concepts, because from this chapter only you will get all the direct questions. Okay, enzymatic activity, there you will get some application type questions, like you have to apply the concept. Okay, there is nothing to worry, it will be very clear. If substrate concentration is less, what will happen to this graph? If temperature is more, what will happen to that enzymatic activity? Like that, some kind of questions with graphical representation you may get. Okay, thank you.